चल कर मेरी ओहदे में लंबी हो गया है ना एमडी हेमेटोलॉजिस्ट ऑन्कोलॉजिस्ट कहाँ थे मैडम अब क्या आधा सुन के क्या करोगे बाकी रील देख रहे हो उसमें आधा हाँ भाई वरुण जी कहाँ था स्टोरी में थोड़ा हल्का उड़ा के भूल जाते हैं चलो कोई भी अच्छा नींबू बताओ कौन बताएगा अभी ये हमारे ने कहाँ जाना चाहता है राइबोसोम उसको जगह दे रहा है जाने का क्या बोलता है तू कौन चलेगा ऐसा but do not worry the DNA has produced another molecule and the name of that molecule is RNA and that RNA is present abundantly inside the ribosomes so RNA recognizes the इकडे तू यार अपना ही मेरा ही भाई है अपन वो दोनों किसने बनाया था चल कोई बात नहीं क्या अटैच हो इसके ऊपर राइबोसोम्स के ऊपर और द प्रोसेस ऑफ हो गया उसका भी जुगाड़ है ना क्या नहीं हुआ अब ये जो तीन मैंने लाइंस बनाई है ये कौन है कोडॉन्स है मतलब एक्चुअली कौन है नाइट्रोजनस उनको किसने प्रोड्यूस किया था मतलब तीन नाइट्रोजनस बेस्ड ऑन द DNA so three nitrogenous based on the DNA are copied in a dash manner on the and they are called as now they are on the ribosome with the help of the now what is the next thing we require actually yes बोलिए what do we require now we require amino acids ayana now we require the amino acids to be arranged in a specific number and a specific sequence will they get arranged yes because अगर ये कौन है पूरा पढ़ो ना भाई तो ये कौन है USC और अगर वो USC है तो कोडॉन कौन सा होगा बच्चों बोलो तुम लोग बोलो कोई A you morons name the amino acid A4 चलो A4 let us take A4 अगर नेक्स्ट होगा तो क्या होगा A2 नेक्स्ट होगा तो क्या होगा नेक्स्ट होगा तो क्या होगा so look at the who is getting arranged according to the sequence of the बोलो nitrogenous of the which have been decided by the nitrogen based on the this is how it works got it that is why we teach you that there are three types of non-genetic RNA one is called mRNA the other is called rRNA tRNA so what is the job of the tRNA अरे वो तो बोलना ही भूल गया मैं what is the job of the tRNA the tRNA will carry the specific amino acids from the cytoplasm to the site of protein synthesis आया ना तो रॉ मटेरियल को जमा करके लेके आने का काम किसका है उसको लाइन में ज्वाइन करने का काम किसका है राइबोसोम पे साइड देने का काम किसका है और ये तीनों की जो बर्थ है सिद्धांत भैया आंखें खोलो जो कहां से हो रही है आया समझ में पीछे के सारे कट्टे अंगूर समझ में आया हाँ बोलो clear everybody pakka all of you now you study now you understood why we study the mRNA RNA and the tRNA because mRNA carries the message for protein synthesis RNA gives a site for protein synthesis of the ribosomes and the tRNA carries the specific amino acid for protein synthesis clear everybody any question चलो out of this, yes, Trupti, what is your question? Huh? No question you have in your life. Now, out of this, from biomolecules, yeah, Raju will tell us. Which is the most abundant RNA? mRNA. Very good. Sit down. rRNA is the most abundant. Almost 8. Hey, Shruti Patil, क्या नहीं तुमको? फैन नहीं चाहिए क्योंकि OLX पे डाल देते उसको मुझे Who is the most abundant one? ठीक है हमारे तो अभी बताया मैंने आप लोग को और you know हमारे नहीं चोगले mRNA is a linear molecule. It's a linear molecule. What do you know a linear molecule? Straight. Straight. You always be straight. Isn't it? Do you agree with you on this? mRNA molecule is always red, or all the RNA molecules are red in which direction? 3' to 5' or 5' to 3'? 5' to 3'. RNA molecules are red in which direction? Shruti Patil, yeah, which direction? And DNA? Okay, it is red in the 5' to 3'. Okay. Easy, no? Very good. But I have already told you, the messenger RNA will have a group of 3 nitrogenous bases. What is this group of 3 nitrogenous bases called? What UAC क्यों बोल रहा भाई? It's called as codon. The messenger RNA begins with a sequence called AUG. Okay, and this is called as the start codon. Correct, everybody? What is it called? Start codon. Start codon. And always codes for an amino acid, methionine or F-methionine. Okay, so with the start codon bachilo AUG, and it codes for methionine or F-methionine. And the messenger RNA also shows the presence of few nitrogenous bases which do not code for any amino acids. Okay, they are called what? Stop or nonsense codons. To be relatable. Okay, who are the stop codons? Bolo. Who are? Bolo bola. Who are? Uga. Who are? These 3 are called as the stop codons or the nonsense codons. Okay, that is UAA, UGA, UAG. Which amino acids will they code for? No. Okay, so they are the ones who will be responsible for termination of protein synthesis. When we will teach you protein synthesis. In my class, following what I am saying? पक्का कि सन्नाटा हो रहा है चलो आ रहा दिमाग में जा रहा है वो समझ में आ रहा है जूली तो ये किसके बारे में था बच्चों mRNA ठीक है later on we are going to study the synthesis of mRNA बाद में पढ़ेंगे by a process which is called as transcription. Okay, then who is the third molecule? That is which RNA? Transfer RNA or the tRNA. Okay. So it has got different models which अलग उसके structures है. So we have a hairpin model, then a clover leaf model, and the L-shaped model. Do you know the clover leaf model, Ruchilo? Good. Then I'll not teach you. है ना? What is the clover leaf model? Explain to me, Ruchilo, one by one. अच्छा नहीं तो ढूंढो. क्लोवर लीफ मॉडल में कितने आर्म्स होते हैं? 3 आर्म्स. कौन से 3 होते हैं, 5 होते हैं, कितने होते हैं? 3 होते हैं. ठीक. एंटीकोडॉन आर्म होता है, एंटीकोडॉन आर्म, टी-साइसी आर्म, DHU आर्म, बराबर है. एक वेरिएबल आर्म, छोटा वाला, और सबसे पहला वाला कौन सा रहता है? वो जो इनकम्प्लीट है, वेरिएबल आर्म वो छोटा वाला रहता है. How do you draw it? Do you draw it like this? The first one. So it's an incomplete arm. How is the arm?
Incomplete.
See, when you say that the arm is complete, it shows the presence of a stem and a loop. So when you said 3 is for the complete ones. They are both stem as well as a loop. But look at the first one. Does it have a stem? Yes. That was an incomplete stem. Isn't it not? So अगर stem loop दोनों है तो उसको हम लोग क्या बोलते हैं कि ये complete है. Stem के यहाँ पे क्या होगा? Base pairing होगा. और यहाँ पे क्या होगा? Unpaired nitrogenous bases. Correct? So this is called as the amino acid. What I have called it? Amino acid acceptor stem, which has phi G and a longer one which ends in CCA3-. It has got a phi G and a CCA3-. And this is the one which will bind to what? To a specific amino acid. It will bind to a specific amino acid. Correct, everybody? Now, what is this arm called? T-psi-C. This one is the variable arm. This one is called as the anticodon. This is the DHU arm. Right? So we are interested in this one more. Which one? Anticodon. Why anticodon? Because it shows the presence of a group of 3 nitrogenous bases which are complementary to the codons on the messenger RNA. So if the start codon is AUG, what is the start codon?
AUG.
Then the anticodon would be what? UAC. Right or no? Anticodon will be complementary to the codons. Got it? So if the codon is CGA, then the anticodon would be GCU. That is why the anticodons are also called NODOC. because they are complementary to the codons. What is called the codons? Did you get this point, everyone? All of you understood what I said? These are unusual nitrogenous bases. T size C. It has got pseudo-uridine and DHU has got dihydrouridine. This protect the tRNA molecule in the cytoplasm. What my point? Because tRNA is found in the cytoplasm. So you don't want RNA breaking down enzymes to come and break the tRNA molecule. Okay? tRNA is also called adapter molecule. It is also called as the soluble RNA. Not because it dissolves in the cytoplasm, but it is very difficult to remove it by centrifugation. That is why it is called soluble RNA. Which molecule chilo? tRNA. Okay. So all this description is not there in the NCERT, but as it is the 12th standard, I have taken results in the formation of RNA. What is the process called? The process is called as transcription. And when the mRNA results in the formation of proteins, then the process is called as translation. So these are the two processes, transcription and translation. All the RNA molecules come from the DNA by what? Transcription. जितने भी RNA है कहाँ से आएंगे? DNA by the process called as transcription. But here we are going to specifically talk about a DNA resulting in the formation of a messenger RNA. Okay. So how will I define a transcription process over here? I'll say that the DNA is a double-stranded molecule. Do you all agree? And one strand of the DNA is information. The information in a strand is copied into the messenger RNA. Got it? क्या बोला मैंने? The information in a strand of the DNA is copied to the mRNA, and that process will be called as transcription. आया definition समझ में? DNA के दो strands हैं, उसमें से एक strand का जो information है, क्या information होता है DNA के एक strand में? वो नाइट्रोजन बेस्ड सीक्वेंस, उसको हम लोग कॉम्प्लीमेंट्री फॉर्म में कहाँ कॉपी करेंगे? और इस प्रोसेस को हम लोग क्या कहते हैं? ट्रांसक्रिप्शन. So at the time of transcription, out of the two strands of the DNA, both of them are not transcribed. Both of them are not transcribed at once. दोनों का भी ट्रांसक्रिप्शन साथ में नहीं होगा, मतलब दोनों यूज नहीं होंगे, कोई भी एक ही कॉपी होता है. And the one which is copied, is called antisense or template strand. What do you call the strand?
Antisense
Or
template strand.
And the one which is not copied is called as the coding strand or the sense strand. I hope you understood my point, everybody. So if I say that, I'll use the two colors only, black and blue. Here I have shown the blue one as the template strand and the black one as the coding strand. So if I say the sequence of the black one, which is the coding strand, is AGC, ATG, CCC. ये किसका सीक्वेंस है? कोडिंग स्ट्रैंड। एग्री करते हो मेरे साथ? ओके। तो आपके हिसाब से बच्चों वो जो ब्लू है वो उसका कॉम्प्लीमेंट्री होगा ना, तो ए के बदले क्या होगा? टी सी जी टी ए सी जी जी जी। ये कौन सा स्ट्रैंड है बच्चों? कोडिंग के एंटीसेंस। ये दोनों स्ट्रैंड में से कौन सा स्ट्रैंड मैसेंजर आरएनए फॉर्मेशन में इन्वॉल्व होने वाला है? एंटीसेंस। तो नाउ द मैसेंजर आरएनए, let me show it in red, will have a sequence complementary to the antisense। तो वही बनाना है ना उसको। तो T के बदले क्या होगा बच्चों? A, C के बदले G के बदले T के बदले A के बदले U, C के बदले G के बदले G के बदले G के बदले। तो अगर आप देखोगे तो आपको ये पता चलेगा कि एमआरएनए जो फॉर्म हो रहा है उसका जो सीक्वेंस है वो कोडिंग स्ट्रैंड के एग्जैक्टली सेम। बराबर है? अगर कोडिंग स्ट्रैंड के पास ए है तो जो एमआरएनए है उसके पास भी क्या है? जी है तो जी, सी है तो सी। सिर्फ एक ही फर्क आएगा, फर्क कि आरएनए के पास थाइमीन नहीं होगा, तो वो हिसाब से जहां जहां उसके पास थाइमीन होगा हमारे यहाँ पे नीचे क्या होगा? यूरिक एसिड। Did your class follow? So now you understood why the strand that is not used is called as a coding strand, because its nitrogenous base sequence is exactly complementary or similar to the mRNA. And the template strand is a strand which is actually copied, that is why वो उसकी क्या रहेगा? Complementary रहेगा। What my point? तो MCQs में अगर कभी ऐसा आया कि डीएनए का स्ट्रैंड है, बोला ये सेंस है और ये एंटी सेंस है और एमआरएनए का सीक्वेंस क्या रहेगा, तो समझ में आया क्या रहेगा? एमआरएनए का सीक्वेंस कोडिंग स्ट्रैंड के एग्जैक्टली सेम रहेगा। जहां जहां कोडिंग स्ट्रैंड में एडेनिन रहेगा हमको एमआरएनए में क्या डालना है? जहां जहां कोडिंग स्ट्रैंड में एडेनिन रहेगा हमको यहां पे क्या डालना है? गाइस मेरा दिमाग है ओके, गिव मी सम लॉजिकल रीजन why both the strands are not copied. I hope you got my point. Aditya, tell me why coding as well as antisense are not producing mRNA. दोनों भी क्यों नहीं बनाते? ये भी और वो भी। Think of a possible reason. The question is for everybody else also. Think of a possible reason. सोच सकता है कोई? Yes, Basil?
दोनों स्ट्रैंड एक दूसरे कॉम्प्लीमेंट्री है, इससे एक का कलर दोनों का कलर सेमी रिजल्ट आएगा।
ओके, एक कैसे बोला दोनों एक दूसरे की कॉम्प्लीमेंट्री है, तो एक का करो या दूसरे का करो सेम रिजल्ट आएगा। ये सही कि गलत है? ये सही नहीं है ना, ये गलत है बच्चों। अगर आप इसको कॉपी करोगे तो एमआरएनए का सीक्वेंस अलग रहेगा, इसको कॉपी करोगे तो एमआरएनए का सीक्वेंस मतलब अभी एक ही डीएनए में से कितने एमआरएनए के सीक्वेंस लिखेंगे? और वो सीक्वेंस में थोड़ा हमारे पास का बेस लिस्ट चेंज होगी नहीं, तो सेम प्रोटीन बनेगी अलग अलग प्रोटीन बनेगी, इसलिए दोनों को नहीं करना। ये पहला रीजन समझा? क्या पहला रीजन? इफ बोथ आर transcribed, what is the problem that will occur? Problem occurs because we will get how many? Two different types of mRNA. Did you get my point? What is the point you got, guys? If you transcribe the black as well as the blue, matlab the coding as well as template, what is the problem that will occur? You will get two different mRNA molecules. So will you get the same protein or two different proteins? Did you get this logic? What is the second possibility? That these two mRNA molecules will be complementary to each other. So there is a huge possibility that the two mRNAs will bind to each other and they'll form a double-stranded RNA. And double-stranded RNA cannot produce a protein. So the process of protein synthesis will stop. So what are the two logical reasons that out of the two strands, only one of them is copied? Because if both of them will be copied, how many mRNA will be produced? So that will form two same type of protein or different type of protein? थोड़ा तो सीक्वेंस में तो चेंज आने ही वाला है, आया ना? दूसरा रीजन क्या होगा बच्चों? कि अगर दोनों mRNA कॉपी हो गए तो mRNA क्या होंगे? Complementary. They'll bind to each other, resulting in the formation of a double-stranded RNA. As a result of which the protein synthesis will stop. Did my class follow this point? That is why out of the two strands of the DNA, are both of them transcribed? Only one is transcribed. And the one who is transcribed is called as a dash strand, template strand, or a, and the one which is not is called or a. Did you get it, everybody? Yes or no? अच्छा, जिसको यूज नहीं कर रहे, जो टेम्पलेट नहीं है, उसको कोडिंग क्यों कहा? क्योंकि उसका और mRNA का सीक्वेंस सेम होता है, सिर्फ थाइमिन यूरासिल से रिप्लेस करना। I have also support. Okay. So that is what is the introduction. How I introduce transcription. It is a process by which one strand of the DNA, which is called as the template strand, will be copied into the mRNA. Got it, everyone? Now see the text. Right? Now do you see two strands, Machalo? Ayana. Okay. One of them is labeled as template, and another one is labeled as coding. Yes or no? Yes? Now, see there, they have written there schematic representation of a transcription unit. लिखा है ना ऐसा वहाँ पे? क्या नहीं लिखा है? क्या लिखा है? Schematic representation of a transcription unit. Yes? क्या नहीं है तुम्हारे बुक में? पूरा पैसा दे दीजिए फिर। क्या मिसिंग है फिर? हाँ? सबसे वो? Now, so where was I? Prachi will tell us. Prachi is very attentive to it, okay? Prachi, you there with us? Okay. What is transcription, Prachi? mRNA को त्रास देता है, बहुत बारिश गिरी तो फिर बह गए mRNA और बारिश गिर गया, बैठ नीचे चल, वो गिरी के ऊपर क्या हो गया, बैठ गई दूसरी चल, So what is included in the transcription unit? 3 things. How many things?
3.
बोलो. Promoter, structural gene, चिल्ला मत, तेज बोल रहा है. मालूम है बहुत खाना खा गया है. तीसरा, terminator. कितने चीज हो गए? टर्मिनेटर सुना आप लोगों ने? तो बोले कहाँ? हाँ? अबे मूवी में सुना था. अभी इतना पुराना पिक्चर देखने का तुमको टाइम मिलता है तो तुम पैदा भी नहीं हुआ था. हाँ? कौन था टर्मिनेटर में हीरो? शर्माई. अर्नोल्ड शिवाजी नगर. कौन सा नगर? बोल कौन सा नगर बोल रही है? देखो 3. पहला वाला कौन सा? First is what? Promoter. Second is what? Structural gene. And third is terminator. Easy. Now, first of all, who is a promoter? अब आधे जनों से the one will promote. Is a promoter. Okay. Don't give dust like answers. See what's the promoter. Promoter is a sequence on the DNA which will help to orient the messenger it will help orient the RNA polymerase in such a manner that it will read the structural gene. कुछ समझ में नहीं आया? देखो. अब this is the which gene बच्चों? Structural gene. Which gene is this?
Structural gene.
Now you want an enzyme. The name of that enzyme is RNA polymerase. To read the structural gene. To read the
structural gene
in such a manner that it will produce a mRNA molecule. You got my point? And this promoter, the promoter, see the NCERT text and tell me, what does the text say? That the promoter is defined with respect to the dash strand. Tell me. The promoter is defined with respect to which strand? Coding strand. कोडिंग और टेम्पलेट अलग अलग है. रीड कर कासिफ के बुक में कुछ अलग है. पढ़ जोर से.
The presence of promoter in a transcription that also defines the template and coding strand.
The promoter defines the template and coding strand. And the promoter is taken on which strand, bachcho? See? It is taken with respect to the coding strand. Promoter is taken with respect to which strand, bachcho? Coding strand. So this is the coding strand. Yes or no? So we take the promoter with respect to which strand? Coding strand. Okay? And the promoter defines whether the strand is a coding strand or a template strand. So if I change the promoter, then the other strand will become coding and जो दूसरा वाला क्या will become the template strand. Did you get this line? I'll tell it once again. This is the coding strand. And who is this sequence, bachcho? The promoter sequence. Pay attention everyone. Which is this sequence? Promoter sequence. With respect to which strand? Coding strand. Now the promoter has decided which strand is the coding strand and which strand is the template strand. If I change the promoter to the other strand, so here now there will be what? Promoter. As a result of which this will become coding and this will become template. So it is the who decides whether it is a coding or a template? Promoter. I hope you got the point, everybody. Yes or no? So who decides whether it's coding or it is a template strand?
Promoter.
The promoter sequence. And the promoter sequence is taken with respect to which strand? Coding strand. Okay? And it will cause transcription downstream. Okay? जहां से promoter start होता है उसके नीचे क्या होगा? ट्रांसक्रिप्शन होगा and that will be called as downstream. उसके आगे वाले region को बोलते हैं upstream. Okay? So the structural gene is present upstream or downstream? ये जो structural gene है ये upstream है कि downstream है? Downstream. ये promoter के आगे वाले region को हम क्या कहेंगे? Upstream region. और ये नीचे वाले region को हम क्या कहेंगे कुछ लोग? Downstream. आई बात समझ में? कौन सा बात समझा? What I meant by upstream and downstream. Okay? So the structural gene is present upstream or downstream? Downstream. Okay? So a transcription unit shows the presence of a promoter, and a promoter is the one which will define. What it will define? The coding strand and the template strand. And then it will be a structural gene which will actually undergo the process of transcription. And the third thing will be what? Terminator is that region where the process will stop. Okay? And you require an enzyme for this whole process. The process of transcription requires an enzyme. And the name of that enzyme is RNA polymerase. क्या नाम है उसका? RNA polymerase. Okay? In the case of prokaryotes, there is only one RNA polymerase. But in the case of eukaryotes, how many RNA polymerases are there? 3 RNA polymerases. Okay? That is RNA poly 1, polymerase 2, and polymerase 3. Okay? So prokaryotes have how many RNA polymerases? 1. And eukaryotes have 3. Who are the 3? RNA polymerase 1, polymerase 2, RNA polymerase 3. And RNA polymerase 2 is responsible for formation of hnRNA, that is heterogeneous RNA, which is a precursor to the mRNA molecule. So the mRNA molecule is formed from hnRNA, that is a heterogeneous RNA, and that heterogeneous RNA is produced by RNA polymerase 2. RNA polymerase 1 will be responsible for rRNA. Polymerase 3 will be responsible for which RNA? tRNA. Okay? You remember that some enzymes are holoenzymes. What is a holoenzyme made up of? 2 parts. What are the 2 parts? Follow? Loudly, Anushka. Apoenzyme plus cofactor. Ayana? Okay. So similarly, RNA polymerase is also a holoenzyme. The main part, okay, is made up of various subunits. The core enzyme part, the apoenzyme part, is made up of many subunits, alpha, beta, such subunits. But other than that, it shows the presence of a factor which is called as a sigma factor. What is the factor called?
Sigma factor.
Sigma factor. And one more factor called rho factor. क्या होता है उसको?
Rho factor.
Rho factor. Okay. So जो अपो एंजाइम है उसके साथ कितने फैक्टर्स अटैच हो सकते हैं? दो. कौन से? सिग्मा फैक्टर और रो फैक्टर. वो जब मिल जाएंगे तब हमको क्या मिलती है? एक कंप्लीट एंजाइम जिसको हम लोग बोलते हैं होलो एंजाइम. और ये सिग्मा और रो फैक्टर का बाइंडिंग जो है उस अपो एंजाइम को ट्रांजिएंट है. हाउ इज द बाइंडिंग? ट्रांजिएंट बाइंडिंग. मतलब the binding is not permanent, the binding is temporary. मतलब the sigma factor can bind as well it can get detached. तो ऐसे binding को हम लोग ने क्या कहा? Transient binding. So how is the binding between the sigma and the main enzyme? Transient. Even the rho and the main enzyme? Transient. You understood the meaning of the word transient, which is? Yes or no? Okay. So look here. This is the single DNA strand. This is another one. Okay, and let me say this region is which gene? It is the structural gene. Okay, and the region आगे वाला will be the promoter. Downstream here will be the terminator. Together it is called as which unit? Transcription unit. Okay. Now the enzyme. What is the name of the enzyme? RNA polymerase will bind to which factor? Sigma factor. And it will help the enzyme. Sigma factor will help the enzyme to recognize the promoter. To recognize the promoter. Understood? So what is the job of the sigma factor? It will recognize the, and then the promoter will orient. It will orient मतलब it will position the enzyme in such a manner that it will be located or it will be looking downstream, so it can read which gene? Structural gene. मतलब ये देखो इस तरफ है ना, अगर ये सब नहीं होता तो कहीं भी अटैच हो सकता था, ऐसे भी जा सकता था वो. तो आरएनए पॉलीमरेज को इस तरीके से अटैच करता है वो कि वो स्ट्रक्चरल जीन को क्या करे? रीड करे, जो कहां पे लोकेटेड है? डाउनस्ट्रीम. मतलब ये आरएनए पॉलीमर जो है वो यहाँ पे इधर उधर क्यों नहीं अटैच हुआ आके? क्योंकि सिग्मा फैक्टर ने उसको हेल्प किया किसको रिकॉग्नाइज करने के लिए? प्रमोटर. व्हाट माय पॉइंट? प्रमोटर के पास और भी कुछ सीक्वेंसेस है जिसको TA बॉक्स बोलते हैं, प्रिमनाउ बॉक्स बोलते हैं. ओके, जो हमारे सिलेबस में नहीं है. तो कुछ ऐसे सीक्वेंसेस है जिसको वो क्या कर देता है? पहचान लेता है और उसको बराबर से क्या करता है? ओरिएंट करता है. ये वाला बात समझा आप सबको? Did you understand the point, everybody? What is the point I just made? कि what is the function of the sigma factor? The sigma factor will help the enzyme. Which enzyme? RNA polymerase to recognize the promoter, which is present downstream or upstream? हाँ? Promoter कहाँ पे? Upstream the structural gene. Yes or no? और फिर वो आरएनए पॉलीमरेज को क्या करेगा? इस तरीके से ओरिएंट करेगा क्या वो रीड कर सकता है. किसको रीड कर सकता है? चलो, स्ट्रक्चरल जीन को. आया बात समझ में? एंड देन द सिग्मा फैक्टर विल गेट डिटैच्ड, क्योंकि उसका काम थोड़ा टाइम के. नाउ लेट अस सी व्हाट द आरएनए पॉलीमरेज डज. इट विल अनस्वाइवल द डीएनए. अनस्वाइवल मतलब खोल देगा. किसको? डीएनए. As a result of which you will see that it has resulted in the formation or it has caused the breaking of the bonds between the DNA, and the two DNA strands have separated in a particular region. Let me call this region as the transcription bubble. क्या कहेंगे मुझे उसको?
Transcription bubble.
ट्रांसक्रिप्शन बबल। ये किसने काम किया बच्चों? RNA polymerase. What my point? And as I've already told you, one of the strand is going to be which strand? Template strand. The template strand will have a sequence of what? Nitrogenous bases. The RNA polymerase will attract complementary which molecules? DNA nucleotides or RNA nucleotides? RNA nucleotides. मतलब यहाँ पे अगर A होगा तो किसको अट्रैक्ट करेगा? U को, G के बदले C को. As a result of which it will result in the formation of a molecule of RNA in complementary to that strand of the DNA. So right now I have a DNA RNA duplex. दोनों साथ में कौन? DNA RNA duplex. इतना सब बात समझ में आप सबको? रिपीट करता हूं। ये RNA पॉलीमरेज ने क्या किया? तुम्हारे वर्ड्स में बोलो, क्या किया? ओपन किया। किसको ओपन किया? DNA को। किया ना ओपन DNA को? तो यहाँ पे कौन सा बबल फॉर्म हुआ? अब ये वाला जो ब्लू लाइंस है वो डीएनए मॉलिक्यूल है कि आरएनए? डीएनए है। क्या है इसके पास? ए टी जी सी सी टी जो भी रहेगा उसका सीक्वेंस। अब ये आरएनए पॉलीमरेज क्या करेगा? उस सीक्वेंस के कॉम्प्लीमेंट्री आरएनए को अट्रैक्ट करेगा। मतलब इधर अगर ए है, जी है, सी है तो सामने क्या लेके आएगा? यू सी जी, और सबको बाइंड करेगा और पॉलीमर बनाएगा। उसका नाम क्या रहेगा? आरएनए रहेगा। आया ना? तो ये रेड मॉलिक्यूल कौन है? आरएनए। और ये ब्लू जो है वो कौन है? डीएनए के स्ट्रैंड के बेसिस है। आया ना? तो अभी क्या हो रहा है? डीएनए और आरएनए साथ में अटैच्ड है ना, तो ये कौन सा ड्यूप्लेक्स हो गया? डीएनए आरएनए ड्यूप्लेक्स। I hope you got my point. So what will the polymerase do? What is the word I am using? Unswivel. मतलब it will open the DNA to form a transcription bubble. Now the RNA polymerase will attract complementary dash nucleotides. RNA nucleotides. Complementary to the DNA template. And it will join the RNA nucleotides and result in the formation of a RNA molecule. But in this region, who is attached? RNA and DNA. And that is called as the RNA DNA duplex. Do you want me to repeat one more time? Will you clear everybody? Did you all follow?
Yes.
Now, as it will keep on reading it, finally it will reach which region, bachlo? T. Who is the T, bachlo? Terminator. And as when it will reach the terminator, जैसे अभी आगे आगे वो रीड करते जाएगा। यस और नो? कौन रीड करते जाएगा? बराबर है? आरएनए पॉलीमरेज आगे रीड करते जाएगा। फिर कौन आएगा बच्चों? रो फैक्टर आएगा कि नहीं? यस और नो? और ये मॉलिक्यूल बनते जा रहा है। कौन सा मॉलिक्यूल बन रहा है बच्चों? 5' 3' RNA. Now with the help of the row factor, it will break what? The RNA DNA duplex. And what will come out is a immature RNA in eukaryotes. It is called hnRNA. क्या होता है उसको?
hnRNA.
That is a heterogeneous RNA. Not a mature RNA. It will be called as a heterogeneous RNA or an hnRNA. In the case of eukaryotes. In the case of prokaryotes, as you have studied in prokaryotic cell, के जैसे जैसे mRNA प्रोड्यूस होगा तुरंत वहां पे राइबोसोम्स भी आके अटैच हो जाएंगे। आया ना? So that they can directly produce what बच्चों? प्रोटीन्स। इसको क्या हम लोग बोलते थे वहां पे? पॉलीसोम्स। याद है वर्ड पॉलीसोम्स? जो प्रोकैरियोट्स में होता है। ये जैसे आरएनए इमेच्योर फॉर्म होता है, डायरेक्टली राइबोसोम उसको क्या करते हैं? ज्वाइन करते हैं और क्या प्रोड्यूस करते हैं, एक पॉलीजोम जिसमें कौन होता है, राइबोसोम्स एंड आरएनए, डीएनए का से राइबोसोम्स एंड आरएनए, करेक्ट है, यस और नो। लेकिन यूकैरियोट्स में पहले क्या फॉर्म होगा, HNRNA, that is heterogeneous RNA, and that heterogeneous RNA will result in the formation of a mature mRNA. Did you get it? Let us see from the NCERT text now. Okay. Let us see. Give me one textbook. Okay. On page number 91 of the new NCERT, old one will be new. Now, see now, line by line, word by word I'm reading. I want your attention now here. Huh? Don't do time pass now. Transcription. Are you there with me, everybody? Huh? Keep your pencils ready if I am putting any point there. The process of copying the genetic information from one strand of the DNA into RNA is termed as transcription. Okay? Look at that whiteboard. क्या लिखा मैंने? Information in a strand is copied to the mRNA. Okay? मैंने पहले बोला था कि जब भी DNA RNA को बनाता है उसको क्या बोलते हैं, ट्रांसक्रिप्शन। लेकिन मैं यहाँ पे किसको बनाने वाला हूँ, mRNA. At the start only I told this. Okay? Next. Here also the principle of complementarity governs the process of transcription. Here also the principles of complementarity governs the process of transcription. मतलब क्या हुआ? What do you mean by complementarity governing the process of transcription? कि अगर DNA का जो टेम्पलेट है उसके ऊपर A होगा तो RNA के पास क्या होना चाहिए, A का कॉम्प्लीमेंट्री, कौन है वो, U. G होगा तो C होगा तो T होगा तो A होगा तो बराबर है। सिमिलरली क्या इसके अलावा कोई ऐसी प्रोसेस थी जो भी कॉम्प्लीमेंट्रिटी से गवर्न थी? Was there any other process which was also governed by complementarity? Lovely. DNA replication. जब हम लोगों ने DNA replication पढ़ा था तो जो टेम्पलेट स्ट्रैंड था, जो आने वाला स्ट्रैंड था, उसका क्या था, सो कॉम्प्लीमेंट्री। हियर आल्सो क्यों बोला है कि व्हाट डू यू मीन बाय हियर, कि इसके पहले भी और इधर भी। यू अंडरस्टैंड द मीनिंग ऑफ द वर्ड हियर? हां या ना? सो व्हेन द NCERT says here also the principle of complementarity governs the process of transcription. So where else complementary had governed? So tell me two places where complementarity is governing: DNA replication and transcription of RNA. Clear? Did you understand the meaning of the line now? If you are wise enough, you would have written with the pencil. Okay. However, unlike in the process of replication, which once sets in, the total DNA of organism gets duplicated. In transcription, both the segments of DNA or only a segment of DNA? And only one of the strand is copied into the RNA. Yes? Have I answered that in the lecture? This necessitates defining the boundaries that would demarcate the region and the strand of the DNA that would be transcribed. तो ये जरूर, इसका मतलब ये होता है कि ये जरूरी हो जाता है कि हम बाउंड्री डिफाइन कर दे कि डीएनए का कौन सा स्ट्रैंड कॉपी होने वाला है। तो वो बाउंड्री हम लोगों ने डिफाइन कर दी कि ये स्ट्रक्चरल जीन है, उसके आगे वाला कौन है, प्रोमोटर है, उसके पीछे वाला कौन है, टर्मिनेटर है। स्ट्रक्चरल जीन कौन से स्ट्रैंड पे होगी, टेम्पलेट स्ट्रैंड पे। यस। Then the question is, why both the strands are not copied during the transcription? Has a simple answer. What is simple answer? If both strands act as a template, they would code for RNA molecule with different sequences. And in turn, if the code for proteins, the sequence of amino acids in the code proteins would be different. Correct? Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Similarly, second, क्या बोले similarly फिर? The two RNA molecules, if produced simultaneously, would be complementary to each other. Hence, would form a double-stranded RNA. This would prevent RNA from being translated into a protein, and the exercise of transcription would become a useless one. Okay? Understood the first paragraph? कल तक याद रहना चाहिए पैराग्राफ. बराबर है? तो ट्रांसक्रिप्शन में दोनों स्ट्रैंड्स एक ही स्ट्रैंड, दोनों क्यों नहीं? क्योंकि दो अलग अलग प्रोटीन बनेगी और आरएनए आरएनए हाइब्रिड बनेगी. ठीक है? ट्रांसक्रिप्शन यूनिट. A transcription unit in a DNA is defined primarily by the three regions in the DNA. What are the three regions in the DNA? Promoter, structural gene, and terminator. Right? There is a convention in defining the two strands of DNA in the structural gene of a transcription unit. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Convention समझ में आता है ना? लोग यूज करते हैं ना convention. कहाँ है convention यूज करते हो? Physics, chemistry में हमेशा यूज करते हो तुम लोग. Since the two strands have opposite polarity, आया ना? 3' 5' 3' 5' opposite polarity होता है. And the DNA dependent RNA polymerase. अब ये RNA polymerase है. कैसे RNA polymerase है? DNA dependent RNA polymerase. Underline that. It's a DNA dependent RNA polymerase. Then also catalyzes polymerization in only one direction. That is which direction? 5' to 3'. Okay guys? कौन सा direction में? 5' to 3'. And the strand that has the polarity 3' to 5' acts as a template and is referred to as the template strand. So DNA dependent RNA polymerase also catalyzes the polymerization reaction only in one direction. Which direction? 5' to 3'. The strand that has the polarity 3' to 5' will act as a template. तो कौन सा वाला बच्चे लोग? ब्लू वाला कि ग्रीन वाला? बोलो ना भाई. Blue वाला will act as a template. Yes or no? In which direction? 3' to. इसे देखो black arrow से पीछे की तरफ गया है. आया ना? Yes or no? And the other hand which has the polarity 5' to 3' and the sequence same as the RNA except that thymine is replaced by uracil is called as the which strand? Coding strand. ये मैंने पढ़ा तो एक बताया था. All the reference point while defining a transcription unit is made with respect to the coding strand. ये भी बताया था मैंने. आया ना? कि हम लोग किसके हिसाब से बात करते हैं? Coding strand के हिसाब से. To explain the point, a hypothetical sequence from a transcription unit is given below. Can you write the sequence of RNA transcribed from the above DNA? Write down with the pencil there. किसने मेरा लिखा इसके सिर्फ क्या क्यों लग रहा है इतना टाइम? लिख लिया ना? क्या लिखा, कैसे लिखा? सेम किसके साथ लिखा? यस सारा नदब, टेल मी क्या क्या लिखा आपने?
सेम का इंस्टेंट जैसा लिखा है।
हाँ
सिर्फ उसमें टी की जगह
तो पहला वाला 3 नाइट्रोजनस बेसिस क्या है आपका? पढ़ो। यूएसी और बाद में क्या आया? ओके बैठ, समझा ना? एग्जैक्टली किस जैसा होगा? कोडिंग जैसा, सिर्फ टी के बदले क्या आता है वहां पे? यू आता है। Then the promoter and terminator flank the structural gene. Flank मतलब on the either sides. Okay. The promoter is said to be located towards which end बच्चों लोग? 5' end upstream आगे की तरफ of the structural gene. The reference is made with respect to the polarity of which strand? Coding strand. क्योंकि उससे पहले ही बोल दिया कि हर चीज किसके हिसाब से होगा? Coding strand. It is a DNA sequence that provides binding site for RNA polymerase promoter, and in the presence of a promoter in a transcription unit that also defines the template and the coding strand. Got it, everybody? By switching its position with terminator, अगर promoter का आपने किसके साथ switch किया? Terminator के साथ, तो बताओ ये सीक्वेंस सेम रहेगा, उल्टा हो जाएगा? वो ऊपर वाली क्या बन जाएगी? कोडिंग, और नीचे वाली क्या बन जाएगी? टेम्पलेट। आया समझ में? Just don't nod your heads. जुई जाई, समझ में आया? क्या समझ में आया? कि अगर हम लोग प्रोमोटर को किससे रिप्लेस करेंगे? टर्मिनेटर से, तो क्या हो जाएगा? और उल्टा हो जाएगा? स्ट्रैंड्स। हाँ ठीक है। The terminator is located 5' or 3'? Upstream या downstream?
Downstream.
हाँ। Now transcription unit and the gene. ये हम लोग बाद में करने वाले हैं। हाँ? Come to the next paragraph. Types of RNA in the process of transcription. Chalo, now I'm tired. I don't want to read it. Who will read it loudly? Yeah, Shruti Patil will read it loudly. Shruti Patil, you got a mic? Not got a mic? हाँ, तो पढ़ो.
In bacteria, there are 3 major types of RNA: mRNA, messenger RNA, tRNA, transfer RNA.
भाई दूसरा लाया तुझे मांगे? हाँ, नहीं ना? पढ़ो बस ना. ढाई लोग दो.
In bacteria, there are 3 major types of RNA: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All 3 RNAs are needed to synthesize a protein in a cell.
बराबर है? आज शुरुआत में बता देना कैसे हो?
The mRNA provides the template tRNA, brings amino acids and reads the genetic code, and rRNAs play structural and catalytic role during translation. There is single DNA dependent RNA polymerase that catalyzes transcription of all the types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription initiation.
Got my point? What is the first step called? इनिशिएशन, जिसमें क्या होता है, आरएनए पॉलीमरेज बाइंड्स टू अ प्रमोटर। ये बताया था ना देखो, फिर वैसे करता है। हाँ, आगे।
It uses nucleoside triphosphate as a substrate and polymerizes in a template-dependent fashion following the rule of complementarity.
Complementarity। हाँ।
It somehow also facilitates opening of the helix and continuous elongation.
हाँ।
Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off.
Young RNA falls off.
So also the RNA polymerase. This results in termination of transcription. And intriguing question is that
Okay, wait. Prachi will tell. Prachi, what do you mean an intriguing question? बोल।
Arising question.
Arising question।
Arising question.
हाँ? Arising question। Okay। आप बताओ। ना बसुन कुछ आ रहा है। Intriguing question हो जाएगा। Who will tell you what is an intriguing question? एक हैरान करने वाला, सोच में पढ़ाने वाला, सोचने में मजबूर करने वाला सवाल क्या है? क्या? डोका? क्या बोला? डोक्याला त्रास। ओके, पढ़ा। हाँ, वाचा।
the intriguing question is that how is the RNA polymerase able to catalyze all the three steps which are initiation, elongation and termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation.
Transiently. Have you used the word transiently? Somewhere have you used it? Transient time.
With initiation factor sigma and termination factor rho to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate.
हाँ, मतलब अगर सिग्मा फैक्टर है तो वो प्रसन्न क्या करेगा? तो प्रसन्न अगर मैं है सिग्मा फैक्टर है तो वो क्या करेगा? क्या? सिग्मा फैक्टर है तो क्या करेगा? लाउडली और रो फैक्टर है तो क्या करेगा? पढ़ो।
In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation take place in the same compartment, there is no separation of
cytoplasm and nucleus in the case of a prokaryote.
Many times, the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. There are at least 3 RNA polymerases in the nucleus. In addition to the RNA polymerase found in the organism, there is a clear-cut division of labor. The RNA polymerase 1
transcribes rRNAs of which type? 28S, 18S, whereas RNA polymerase 3 is responsible for transcription of tRNA, 5sRNA, snRNA, small nuclear RNAs. And which one? RNA polymerase 2 is responsible for hnRNA, that is heterogeneous nuclear RNA. Second complexity we want to see it tomorrow. Okay, but are you clear with today's part total? So क्या क्या आपका भी इसका सारांश है? सबसे पहले ट्रांसक्रिप्शन का डेफिनेशन सबको समझ में आया? डीएनए के दोनों स्ट्रैंड के एक ही स्ट्रैंड ट्रांसक्राइब हो रहा बराबर है, और वो जो एक दोनों क्यों नहीं होते ये समझ में आया? उसे कितने रीजन बताया? तो मेरे ख्याल से समझ में आ गया आपसे, ठीक है। उसके बाद ट्रांसक्रिप्शन के लिए आपको डिफाइन करना पड़ता है ट्रांसक्रिप्शन यूनिट, बराबर है? अच्छा, अब म्यूट करा दो। जो